Okay, so we're on to stage 3, and just as a recap, as we work through each of the stages, it's probably helpful to keep coming back to this major process that just shows all how they're all related and the entire process of cellular respiration from start to finish. So remember, we started with glucose. We break glucose down in a process called glycolysis. The product of glycolysis, which is pyruvate, travels into the mitochondria, right into the matrix, so the very, very middle part of your mitochondria, and it becomes oxidized. And so during that process, we remove some carbon dioxide and we form something called acetyl-CoA or acetyl coenzyme A. And so it's this product from pyruvate oxidation that drives the start of the next part of the cycle. So remember that stage 2 only happens if we have oxygen present. Remember that if we don't have oxygen present, after glycolysis, we go to lactic acid fermentation, or if you're a yeast, alcoholic fermentation. So oxygen's present, so we undergo pyruvate oxidation, and we form this that becomes the starting material for Krebs cycle. So now we're going to look at Krebs cycle in a little bit more detail. Here's the overall main purpose or summary of it. It further breaks down pyruvate to make ATP. So we get a little bit more ATP coming out of the Krebs cycle. We're going to see how much in a minute. It again transfers some electrons to NAD and FAD. So FAD I don't think we've seen yet either, but it's also another coenzyme that is handy in transferring electrons. So it's involved in redox reactions just like the NAD. So we're either adding or removing hydrogens from these to transfer electrons, which you'll see by the time we reach stage 4 become really, really important. So there's things that we're learning along the way that won't necessarily make complete sense until you've got the entire process from start to finish. So that's why we can go through this in steps, but it'll be definitely beneficial for you once we've got all of them done to go back, and then you'll be able to even draw more links between these stages as we go. So in Krebs cycle, we actually also remove more carbon atoms in the form of carbon dioxide. So remember our final product, and this is our real life example, not scientific, that when we breathe in oxygen, we breathe out carbon dioxide. Here is our source of carbon dioxide. This is going on in our bodies at all times, as long as we are breathing oxygen in. Okay, so we got a little bit of carbon dioxide off of the pyruvate oxidation, but you're going to see that we get a lot more carbon dioxide out of the Krebs cycle. And it also occurs in the matrix, just like pyruvate oxidation. And again, I already stated that you have to have oxygen in order for this process to continue. Okay, so I gave you guys a fill in the blank, and I think we'll start with this one first. Just a summary. We already talked about pyruvate oxidation, so I'll skip down to the citric acid cycle. So there's one perfect thing that you guys probably would like to know. The citric acid cycle is another term for the Krebs cycle. Okay, and I'll show you why in our first sentence here. So acetyl-CoA enters the cycle. So remember, this is from our pyruvate oxidation, and it combines with a compound called oxaloacetate. And what you make out of that is a 6-carbon compound called citrate, also known as citric acid. Okay, and that's where the citric acid cycle is sometimes used instead of Krebs. Krebs is obviously named after the scientist who studied this cycle in detail. So Krebs cycle or citric acid cycle has 8 steps, and it's this molecule here that undergoes a number of reactions in order to release some carbon dioxide, release some more ATP, and we eventually convert citrate into our final product, which was also our starting product, oxaloacetate. So now you can probably see why most representations of the citric acid cycle are in circles. And it's in a circle mostly because it's a cyclical cycle. What we start with, we try to aim to finish with, so that that cycle can continue. If we're getting more acetyl-CoA coming in, then we need the oxaloacetate there in order to make citrate. So the final product or outcome of citric acid cycle is that starting material as well, so that it can be used again and again. So there's another example of recycling. So here's a summary of our main sort of products released throughout this cycle. So we get carbon dioxide released, and they say waste because it's something our body breathes out and gets rid of, or we expel it. And we end up with some NADHs and FADH2s. These are really important for the final stage of cellular respiration. So this whole thing here may not make a whole lot of sense until you see what their job is in the last stage. All you need to know right now, though, is that they're produced. So energy is released in the form of ATP. Glucose molecules produce 2 molecules of ATP because 2 molecules of pyruvate are created from each molecule of glucose. So let's look at this cycle in a little bit more detail. So here we have our acetyl-CoA. This was produced from pyruvate oxidation, so this becomes our starting point. Acetyl-CoA enters our cycle, okay, and the coenzyme A actually comes off, and it's just the carbon, hydrogen, and oxygen portion of acetyl-CoA that continues, joins with another molecule called oxaloacetate, which is here. Maybe we should put that in there so that you can actually see. I don't know if you guys did that or not, but let's do that. So this guy right here is our oxaloacetate. So this here is oxaloacetate. So you can see acetyl-CoA comes in from the pyruvate oxidation, combines with oxaloacetate, the coenzyme A comes off, and you're left with this bigger molecule. So we end up with this molecule called citrate. So this is citrate here, okay? And from this stage to the next stage, all it has done is it's rearranged its atoms. So if you recall from Unit 1, our biochemistry unit, when you have the same number of elements, but just different formation or different arrangement, we call that an isomer. So we start off with citrate, we shuffle some things around, but we don't actually change the total number of atoms present. It's called isocitrate because it's an isomer. So, okay, same number and type of atoms, just a different arrangement. Next stage, CoA is coming back in, okay? And you're going to see that we are reducing NAD to NADH, and we're releasing some carbon dioxide as waste that came off of this isocitrate. And I've highlighted here what happens with these, okay? Remember, these are oxidation reduction reactions. And I stressed that they were important to the final stage, which is stage 4. So you should know, if I were to give you, say, like a blank copy of this, what's happening in these little bubbles here, that we are reducing NAD to NADH, carbon dioxide's coming off. Again, and you're going to notice that each of these are the same thing. We're reducing NAD to NADH, we're reducing FAD to FADH2, and we are reducing again here. And each time, the products of those, so the NADHs, are actually all going to go to stage 4. So you might want to add that to your notes as well. They are produced here, but they play a bigger role in the final stages. So you don't need to know the names of each of these molecules. What you should know is the first one, oxaloacetate. It's also the final one. So we start with this, we finish with this. You should know that acetyl-CoA is coming in to start off the beginning stages of the reaction, and you should know that it produces citrate here. After that, we are not concerned with the chemical name or the structure of these, so don't worry about remembering those, but just remember what overall is happening to these molecules as we progress, that we're reducing a bunch of these coenzymes here, okay? And at one stage as well, we are converting ADP back into ATP. So we make 1 ATP per 1 cycle of this Kreb cycle, okay? And we make 1, 2, 3 NADHs and 1 FADH2 for every turn of this cycle. So those are really the important things you should know. Maybe we can just highlight. So we should know the starting materials are these 2 things. Okay, we should know once those 2 starting materials combined, what does it make? And the reason we should know that is because it helps determine what the name of this cycle is, the citric acid cycle or the Kreb cycle. Okay, and then we should know what's happening here in each of these places, because it's the same thing that's happening each time. We're reducing these enzymes. Okay, and then the final thing you should know is that we make 1 ATP throughout this cycle.